So hi, hello and welcome again. Microbehunter here. Well, today we're going to have a very deep look into a cell, into this paramecium here. This paramecium is a single-celled microorganism. It's a protist. And I would like to explore the question today is, why does it not explode? Why does it not pop open? Right? Now you might wonder, why should it? Well, that is the whole point here of this video, because it always has water moving into the cell. Those Paramecia, like many other cells, continually take up water because water flows into the cell by osmosis. And actually, this should cause the cell to burst and to pop open. However, this doesn't happen. So evidently, the Paramecium has some kind of a mechanism in place to prevent it from bursting. And as a matter of fact, we can actually see this quite well under the microscope of what it is able to do to prevent it from bursting. And yes, maybe you've already seen that inside the cell, there are these two large circles that are always keep on changing their size. I'm going to pause this video here. It's not a live image that I'm showing you here, but actually it is a video because it does give me a little bit more control over the whole thing. Do you see this little round structure over here and over here as well? These are referred to as contractile vacuoles, and I would like to simply ask you to simply have a look of what they do. You're going to see it's almost they're pulsating. They're changing the size, and as a matter of fact, they work like pumps that pump the water out of the cell again. And later on, I'm going to show you in phase contrast a very large magnified contractile vacuole, how it's actually working in action. I think quite nice to see. But let's zoom in a little bit more here. And this here, again here on the side, this is the contractile vacuole over here. Now much bigger. Therefore, the video is a little bit blurry. But you can see that actually on the side here, maybe carefully, you're able to see those 5 or 6 other canals surrounding it, almost like a star shape. These are the canals that collect the water and actually direct the water into the contractile vacuole. Now it pumped it out again. See, it collapsed now, and now it's slowly filling up again, filling up with water again. And then this contractile vacuole actually has a little opening to the outside of the cell where all of the water is pressed out again. And this is basically the way how the paramecium protects itself from bursting, because on the surface of the cell, there's the cell membrane, and water is always going into the cell by osmosis. So I would like to also show you now the surface of the cell itself. Okay, you see that there are these tiny moving here. These are called cilia on the surface. And those cilia, they constantly beat. You can see that there are also some bacteria and some other organisms floating around here very fast. This is not time lapse. This is real. In real time. And those things that you see inside the cell here, these are that's food vacuoles and so on. In the contractile vacuole, see, it's a little bit, I'm covered up a little bit here, and it's actually here on the side. And by osmosis, there's always water flowing into the cell. The cell, of course, is expanding, but the cell membrane on the outside is not strong enough to actually hold the cell together, and for this reason, you've got this contractile vacuole over here that pumps the water out again. I would now like to show you the video that I made with phase contrast, because even though it looks a little bit blurry, you can see the procedure much better. Okay, and over here again, look here it is, the contractile vacuole in the center. It starts to fill up with water. Right, it looks now quite bright. You see those 1, 2, 3, 4, 5 supplying canals around it. And then again, when it's full, it pulls together again and it pumps the water out. It's quite fascinating. And again, the whole thing is a little bit blurry because I really zoomed in quite a bit, and but still we're able to see it better this way. And last but not least, here now I really made it big. Look, this is it. That's the contractile vacuole, and see how it pulls together again. And how it therefore is able to expel all of the excess water. Now, how is it able to do that? Well, there are so-called microtubules. These are protein fibers surrounding the vacuole, the contractile vacuole, and those microtubules, they pull together and they compress the vacuole, the contractile vacuole, and this causes the water to be expelled out of the cell again. Now, I think it's always quite fascinating to see all of these processes of life so well under the microscope. If you like this video, then of course I would like to invite you to subscribe to this channel. But for today, I think that's all I wanted to share with you. I wish you all the best. Happy microbe hunting as always, and see you around next time. Bye-bye.